Okay, so in this video, we're going to be talking about the actual format of a lab in AP Biology. So these labs are going to be broken up into two parts. The first part is the part where it's largely teacher directed. Okay, so that first section involves a pre-lab where we sort of plan everything that we're going to be doing. And then we do data collection where we actually get the actual experiment going and we collect data. And then we do our data analysis. And this might involve drawing graphs, and it also often involves doing some math or statistical analysis. After that comes the more student-directed part. So this is student-directed because it's now your opportunity to go ahead and make the lab your own, to investigate something that you're interested in, to make changes to the lab in order to use the same techniques and study the same general topic, but to learn something that we might not have learned if I just took command of everything. And this lets you practice actually being a scientist instead of just being a science student all the time. After that, you have to go ahead and write a lab report, because if you're going to be a scientist, you got to publish your results and tell me what you learned in your student-directed part of the lab. You can see why these take a long time. We basically do every lab twice. And so these labs take on average about 6 days, and some of the labs actually take about a month while we're growing plants and growing several generations of plants even in some of the labs. So the first thing we do is a pre-lab where we kind of go over background information. So the lab that I'm going to be explaining this time is one where we're going to be using these little guys, sometimes known as roly-polies. We're going to be calling them pill bugs, even though their official sciencey name is that they are isopods. And so I'll give you some background information about them just so that you get comfortable with them. And then I'll also talk about our apparatus during the pre-lab lesson. And in this case, our apparatus is a choice chamber. This is two different environments that are connected together by a little passageway. So the pill bugs will go in here. They'll either go in the dry condition side or the wet condition side. And they'll get to move back and forth at their choice. So if this allows us to see if pill bugs prefer a dry environment, or if they prefer a wet environment, or if they really just don't care. And so we're going to be actually analyzing, our big word is we're going to be analyzing behavior, specifically the chemotactic response. Okay, so organisms that like a chemical, let's say it's little pill bugs like this, and they are attracted to chemical X. When they are attracted to that chemical, we say that they have a positive chemotactic response, and that might be like butterflies that are attracted to nectar, because that's where they get their food. If organisms move away from a certain chemical, let's say this is chemical Y, then they have what we call a negative chemotactic response. And so we're going to see if pill bugs have a positive chemotactic response to water or a negative chemotactic response. In order to actually design our experiment, we need to identify what are the different variables from this situation. And variables are anything that you can change about a situation or anything that you can measure. So there's lots of things I can measure. We'll come up with huge amounts of ideas for this. But I'm just going to pick a few, so we might pick like the size of a pill bug. Right? We might pick the species of bug. We might realize that we can change the environments of the chamber, so we can change like the wetness of the chamber. We might find that we can change the sugar amount or the pH amount in each chamber. We also might find that we can measure the number of bugs on each side, or the number of bugs per side. Okay, so once we've got this long list of different variables, we're going to sort these variables into things that we control. Things that we control are called independent variables. And the things that we control are, for example, the size of the bug is an independent variable. The species, the wetness of the environment, the sugariness of the environment, the pH. One thing that we do control the number of bugs per side at the beginning, and then we're going to be looking at dependent variables. Dependent variables are things that change or that might change because of the independent variable. So we don't expect that changing the size of the bug would change the sugariness of the choice chamber. So sugariness is not a dependent variable. Neither is wetness or species. The only thing that's dependent on this list is the number of bugs per side, because everything else we directly control. We don't control the choice that bugs make. We don't force them to move to one side or the other. All right. So once we've got our list of independent and dependent variables, we're going to pick the ones that we'll actually investigate. In this case, we're going to investigate wetness. We only get one independent variable, and there's only one dependent, so we'll see if that affects the number of bugs per side. Okay? So these are our variables we're going to examine. Everything else, size, species, pH, all of those other variables become what we call control variables. So wetness will change in our experiment. We'll change it from one test to the next. Dependent variable, the number of bugs per side, is what we're going to measure for each experiment. And control variables are ones that are going to not change from one to the next. So you want to make sure you've got the same species of pill bugs every single time. Okay. Next, we move on to actually carry out the experiment, and we do data collection. In the data collection section, we'll be taking observations. A lot of times we draw scientific sketches like the one to the right. You'll notice that scientific sketches require us to label details. They require us to draw carefully rather than just drawing a little dot with legs. You know, this is not a scientific sketch. Okay, even though it might fly in your little cartoons. And then we also create data tables like the one on the bottom. So you can see that in this one we've got the control test. This one has both side A and side B were dry, so there's no wetness in this at all. And then this is the experimental, where side A was wet and side B was dry, so it actually mattered. Sorry, actually that's the other way around. So side B was wet in this one. After we've done our actual data collection, we go to data analysis. So we're going to draw a graph. We do two different kinds of graphs in this class. We do scatter plots if both the independent variable and the dependent variable are numbers. We'll do bar charts if both the independent variable and the dependent variable are not both numbers. So it actually should be I or D is a number. Okay. So in this case, our independent variable, by the way, your independent variable always goes on the x-axis. Our independent variable is the wetness, it's the environment. So I'm just going to look at the experimental group here. So we had like the dry side and we had the wet side. Okay, and then our dependent variable is the number of pill bugs. Now dry and wet are not numbers, but number of pill bugs is a number, so we're just going to be drawing a bar chart, and it might look something like this. So you can see there were a lot more bugs on the wet side than on the dry side for the experimental test. The other thing we do a lot of times is statistics, because you might ask, well, sure, there were more bugs on the wet side than the dry side. And that might mean that pill bugs prefer wet environments, but it could also just be random chance. So in science, we have to prove, especially in biology, we have to prove that animals aren't just behaving randomly, that they actually prefer the wet side to the dry side. And that requires some mathematical analysis, and we'll learn how to do that in the next video. Lastly, having finished our analysis, we'll do our conclusion. We're then going to move on from that conclusion to ask another question. We might have discovered that these pill bugs do prefer wetness. And then we might ask the related question of, do fruit flies prefer wetness? Does it apply for other small organisms? Not necessarily related. You know, the fruit fly is an insect and the pill bug is actually not an insect. But, you know, is there some relationship there? And so then during the planning phase, you have to write your own procedure, and then you have to carry out your own lab going through all the parts: pre-lab, data collection, data analysis, so on and so forth. And that's what we're going to be doing in the next part. In the next part, I'm going to show you how I would write my lab for the pill bug lab. And then you're going to go on, in order to practice, writing your own lab using data related to fruit flies.